Hi friends, I am Vaibhav from Vaibhav E-Learning Academy. Today we are going to start a topic that is cell cycle and cell division. This is our part number 1. So in today's video we will be discussing about what is cell cycle and what are the methods of cell division. So every organism starts from a single cell called zygote, and from a zygote, from small to a big, the organism grows. But how does this grow? The organism grows because of the multiplication of the cells. The cells multiply and then the body starts to grow. This is why the body grows, and the reason is due to the cell division. Now what are cell cycle and what are cell division? Cell cycle is the cycle in which the cell prepares itself for the cell division. So in this cell cycle, the cell grows and the genetic material synthesis takes place, and these things happen inside the cell during the cell cycle. And during the cell division, the cell divides, that is the cytoplasm of the cells divide in this cell division. So cell cycle and cell division. So let us start with the cell cycle. This cell cycle has 3 major phases. First one is G1 phase. Second one is S phase, and the third one is G2 phase. G1, S and G2. Here G means gap and S means synthesis. So the gap phase and the synthesis phase. So G1 is equals to gap 1 phase, S phase is synthesis phase, and G2 phase is the gap 2 phase. So what happens in these phases? These are actually coming under one part. This is like one circle over here. From here and something like this. So here is the G1 phase, this is the S phase, G2 phase, and this is the M phase. What is the M phase? M phase is the mitotic or meiotic phase. So here G1 phase, G1 in the sense gap 1 phase, S means synthesis, and G2 means gap 2 phase. So this G1 and S and G2, these all 3 come under interphase. Now what is this interphase? The interphase is the time, like I said, the time required for the cell to grow and the genetic material to synthesis the genetic material. So that is the time in the interphase, and M phase is the time when the cell actually divides. So starting with what and all happens in all these 3 phases. Here, first one, in G1 phase, which is the gap 1 phase, what happens is nothing much happens, just the cell starts to grow. For example, this is the cell. In G1 phase, this starts to grow a little bit like this. Here, no synthesis of DNA takes place, no duplication of DNA, no duplication of chromosome, nothing else happens. Only in the G1 phase, the cell increases its size. Now, in the S phase, this is the synthesis phase. In the S phase, you can see the synthesis of DNA. Now, what is the meaning of synthesis? Synthesis means production, but here production in the sense duplication of the DNA. Synthesis of the DNA, it means that already there are some amount of DNA present inside the cell, but they duplicate it. So DNA is a double-stranded like this. They will duplicate the number of the DNAs, so that both the cell after division, that this one cell will become this and this. So both get equal amount of DNA. That is why the DNAs are synthesized, that is duplicated. These things happen in the S phase. And in G2 phase, we have synthesis of proteins, and proteins majorly histone proteins. Synthesis of proteins, majorly histone proteins. So G1, S and G2 phases, these 3 are the main phases. Now along with these 3 phases, there is one more phase which is called G0 phase. What is this G0 phase? This G0 phase is called quiescent phase. Now, what is this quiescent phase? there are some cells which enter the G1 phase. Entering the G1 phase in the sense the cell size is increased, but they do not enter the S phase. So they are stuck between G1 and S phase because they are waiting for the appropriate time to divide themselves. For example, liver cells. The liver cells divides in every 1 or 2 years. So they remain in G0 phase or the resting phase. So in this phase, quiescent phase, they stay between G1 and S phase. So they are over here, G0 phase. Why? So because the size of the cell has been increased, but it is not entering the S phase. When it needs to divide itself, then it will enter the S phase. So these are the main phases of the interphase. Now, these three phases together are called as the interphase. Interphase is called as the resting phase, the phase in which the cell is preparing for the division. This M phase, here M can be of two meanings. One can be mitotic and one can be meiotic. Mitotic phase or meiotic phase. Mitotic, when we are learning about mitosis, then this is mitotic phase, and when we are learning about meiosis, then it is meiosis phase. So, the cell division occurs in humans. The cell division occurs once in 24 hours. In humans, the cell division occurs once in 24 hours. In yeast, it occurs once in 90 minutes. In 90 minutes. So this cell division it does not need to be constant. It can depend upon many different values or many different factors also. Even in humans, when the human is young, when there is a young child, the cell division rate is more than that. Cell division rate in the sense, still the cell division happens once in 24 hours only, but the speed is fast. In the medium, like in the adults, in the teenagers and adults, the cell division rate is normal, like it is low. So it occurs once in 24 hours, but too many number of cells do not divide. And in olden age, the cell division speed or rate is very low. It still happens once in 24 hours, but the rate is very, very slow. So that these are many factors which depends upon the cell division. So cell division depends upon these all factors. In humans, it is once in 24 hours. In yeast, it is once in 90 minutes. So these are about the cell cycle. Now cell division. Now here cell divisions are of two types. One is meiosis and one is mitosis. So first one, mitosis. So before mitosis, any cell division has two parts. One part is called karyokinesis and one part is called cytokinesis. Karyokinesis and cytokinesis. This karyokinesis, in here cell division, I am talking about mitosis now. Karyokinesis has four parts. First part is prophase. Second part is metaphase. Third part is anaphase. And the last part is telophase. Here karyokinesis means division of the nuclear membrane. Here division of the nuclear membrane in the sense division of the organelles and organelles inside, the division of the nuclear membrane, the division of the genetic materials inside. So these are all happen in karyokinesis. Cytokinesis is the division of cytoplasm. So cytokinesis is the division of cytoplasm. These do not have many different steps. So this karyokinesis has four steps over here: prophase, metaphase, anaphase and telophase. Now in mitosis, let us see what happens in each and every phases. Mitosis, this is called equational division. Equational division. Equational division in the sense, mitosis, there is the parent cell. This 2n means it is diploid. After the cell division, the daughter cells are also 2n. These are also diploid. So, equational division in the sense that the number of chromosomes, that is the 2n over here, this number remains constant. Here also it is diploid and here also it is diploid. Diploid in the sense 2n. So, in our body, the somatic cells are called the body cells. So, somatic cells are called body cells. So, body cells, and there are one more type which are called germinal cells, germ cells. Body cells are 2n and germ cells are single n. Body cells are the cells which are present in the body and germ cells are the sperms and ovum. So these two cells are the germ cells. Body cells are always diploid, that is 2n, and germ cells are always haploid. So this is what here 2n is diploid, and mitosis does the equational division. It means that the diploid number is maintained even in the daughter cells. This is what mitosis is. Now the first phase is prophase. What happens in prophase? So let us take a cell over here, and here is the nuclear membrane. Okay, so better let this be the nuclear membrane and this be the cell. This is the cell membrane, this is the nuclear membrane. So in prophase, what happens is, there are two phases. First phase is early prophase, second phase is late prophase. So in prophase A, let us take early prophase. So we all know that inside a nucleus, for example, this is nuclear membrane. Here, this is the nucleus, totally nucleus, nuclear membrane, nucleoli, and these are the chromatin threads. Chromatin fibers. So these chromatin fibers are tangled inside the nucleus. But at the early prophase, this tangledness, they start to condense. So the first point you can say, the chromatin fibers start to condense. The chromatin fibers start to condense. Now, after condensing, they form structures like these. So what are these? These are called chromatids. So these are called chromatids. Now, during the prophase, early prophase, what happens is the chromatin fiber starts to condense, and second point is the organelles. Organelles. Here there are so many organelles, right? Like the endoplasmic reticulum. Like from here you can see like this. Endoplasmic reticulum, the mitochondria, and then many small granules. So these all start to disappear. Start to disappear. These two things happen in the early prophase. B, late prophase. In late prophase, let us take the same cell over here. Now here in late prophase, what happens is the nuclear membrane disappears. So this nuclear membrane disappears like this, and still the chromatids are moving over. The chromosomes which have been condensed, the chromatid fibers which are condensed, are still moving over here. So they are moving randomly. The second one which happens over here is all the organelles have disappeared. Now you may wonder, have disappeared. What is the meaning of have disappeared? So disappear in the sense they get dissolved inside the cytoplasm because they are made up of proteins and they can be formed again later on. So all the fibers, like including the ribosomes and the granules, the vacuoles, mitochondria, Golgi complex, endoplasmic reticulum, everything gets dissolved inside the cytoplasm because they are made up of proteins. So these are the things which happens in prophase. In prophase, what happens? In early prophase, the chromatid fiber starts to condense, the organelle starts to disappear. Then in late prophase, the nuclear membrane disappears and all the organelles have been disappeared. So this happens in the prophase. Now second one is metaphase. What happens in part over here is actually metaphase. Second one, metaphase. So metaphase is one of the most important phases in the cell division. So in the metaphase, what happens? This is the cell. Now the nuclear membrane has disappeared, so no need to draw that. So these are the chromosomes. So first, what happens in metaphase is all the chromosomes are first roaming here and there, right? Chromatids are moving here and there. This is before. But in metaphase, what is the main thing that happens? All the chromosomes or chromatids lie on the metaphasic plate in a certain order. So what is the certain order over here? The chromosomes combine, the chromatids combine and form chromosomes. So over here, what is the metaphasic plate? Metaphasic plate is the equatorial plate which is over here. So this you know what is equator, right? Equator is the plate which is over in the middle, that is called the equatorial or the metaphasic plate. So here they are arranged like this. So like this, there are 26 chromosomes, so they are arranged, 26 chromosomes are arranged. This is one thing. The second thing is the aster rays start appearing. What are the aster rays? The aster rays starts appearing. Aster rays are, these are the centrosomes. So inside the centrosome there are centrioles. So over here, one centriole is over here and one centriole over here. You know what are aster rays? The aster rays are the rays which are formed towards the back side. These rays are the ones which initiate the cell division. So these rays are called aster rays. So the aster ray starts appearing. This happens in the metaphase. So what happens in the metaphase? All the chromatids lie on the metaphasic plate in a certain order. They form chromosomes like this and the aster ray starts appearing. So these all happen in the metaphase. Now after metaphase comes anaphase. The third phase over here is anaphase. What is anaphase? So here the centrioles are cylindrical structures. Over here there are the chromosomes. Okay. And these are the aster rays. So in anaphase what happens is, first step, there are two steps that happen in the anaphase. The first step that happens in the anaphase is that the so the rays come from the centrioles and attach to the chromosomes. Chromosomes at the point, the spindle fibers appear called the chromosomes. And the second phase is the spindle fibers pull the chromatids to their opposite poles. So it means here, what are the spindle fibers? The fibers which come out from the centriole, they come and join here. This is the first phase, what happens, and after that what happens is they get separated, they get pulled. So over here, if there is a centriole, they are pulling it. These pull it towards themselves, these pull towards themselves. So this is the chromosome, that is the genetic material is being transferred. The spindle fibers appear and hold the chromosomes, and the spindle fiber pulls the chromatids to the opposite poles. Here they hold the chromosome, but they are splitting it into two parts. That is why we say chromatids instead of the chromosome. Now, the last phase is the telophase. Fourth phase, telophase. What happens in the telophase? So first thing, again here is the cell. The chromatids move to the opposite poles. The nuclear membrane reappears, and C, organelles reappear. So this time the cell will not be like this, but the cell will be little bit big in the size, and here the nuclear membrane have been reappeared again. So this is again the nuclear, and one more thing happens. The chromatids again form chromatin fibers. So again the chromosomes, they form the chromatid fibers. All the organelles reappear. This is the nucleolus. Chromatid fibers are formed once again, and then all the other organelles are formed. These are the granules, then the Golgi body, the mitochondria, and then the endoplasmic reticulum. Everything is formed back. Everything is, all the organelles, and the cytoplasm is also done. So this is that the cell division. Part A is done. Part A is the karyokinesis. The nuclear membrane and the genetic material have been divided. Now the last one is actually cytokinesis. Now the cells have been like the nucleus, the nuclear membrane have been formed upside and downside. Now we have to make this one cell into two cells, right? That is done by cytokinesis. Now is the main part, that is cytokinesis is the division of the cytoplasm. So over here that the cells, both the cells, one part has been up, one part was down. So cytokinesis, what happens is cleavage furrow. This is a term, cleavage furrow. What is cleavage furrow? Cleavage furrow in the sense, now over here, this is one big oval over here. Here nuclear membrane, here nuclear membrane. Cleavage furrow in the sense, now something will start happening in the middle. This will go like this. This portion over here is called cleavage furrow. This one is also over here called cleavage furrow. Slowly this again goes inside, this also goes inside, and at one point this becomes one cell and this becomes one cell. So this is what happens in cytokinesis. This is how mitotic cell division takes place. The daughter cell, both the cells, they have same diploid number of chromosomes because the chromosomes were duplicated and been sent. So that is it for today's video. In today's video, we learned about mitosis, what happens in karyokinesis, what happens in cytokinesis. In the next video, we'll learn about meiosis. We'll learn about the differences between mitosis and meiosis. So see you next time. Do like this video, subscribe to this channel and share this video. See you next time in the next video. Thank you and goodbye.